Isn't it amazing? I had to come up to you. I think we're co-authors. And some. I'll get it. No, it's 21.
All right, guys. Happy Thursday, everyone. Time for good news. Lots of it this week. So we'll start off with Dr. Dylan's lab, who pops up quite a lot on Thursdays. Her graduate student and postdoc both got abstracts accepted for presentation at the American Society of Nutrition. So Agnieszka is presenting a poster on a cross-sectional analysis, or a presentation at least, cross-sectional analysis of food security, race, and sex on cardiometabolic and cognitive health outcomes in young adults with overweight and obesity. And Ayodeji is also presenting on the impact of almond consumption on adipose tissue gene expression profiles in adults with obesity. So congratulations to that. Last week on campus, we had Cardiovascular Day, and we had a great representation from NEP. We had a number of winners, including Neil, who won, Neil and myself, who won posters for graduate posters, and Natasha, who won the postgraduate poster award, and Dr. Larissa Dos Santos, who's in John Padilla's lab, also won the Harold M. Laughlin Award. So that's a really great representation for many people. Finally, I'd like to congratulate my PI, Dr. Jean Padilla, who's been selected by his peers to receive the 2024 Plattner Outstanding Mid-Career Research Award. Congratulations to the CV Day winners and Dr. Jean Padilla.
Those of you who don't know me, my name is Elizabeth Parks, and I'm a professor in nutrition and exercise physiology. And I'm here to introduce Dr. Marc Cornier. He's a physician scientist with expertise in obesity and metabolic disorders. He graduated with his basic science degree, Bachelor's of Science degree in engineering from Vanderbilt, and then went on to medical school at the Medical College of Georgia. Following graduation, he performed his internship and residency at Georgetown University, and then went on to do a fellowship in endocrinology at the University of Colorado, Denver. At Colorado, he got hired, and then he rose from being an instructor to assistant to associate to full professor, and he held several leadership positions. He was director of the Advanced Lipid Disorders Clinic, and he was also associate program director for the Obesity Medicine Fellowship, which is very well known at the University of Colorado. In 2021, he was allured to the Medical University of South Carolina in Charleston, and he became the director of the Division of Endocrinology, Diabetes, and Metabolic Diseases. And he was named the James A. Keating Endowed Chair. Nationally, he served multiple professional society committees, including chair of the Clinical Endocrinology Update Steering Committee for the Endocrine Society, and the chair of the Obesity Committee for NPAM, the Nutrition, Physical Activity, and Metabolism Council of the AHA. He's currently serving as president-elect of the Obesity Society, so he will be very busy next year. Dr. Cornier has been funded by the NIH, and he's currently the PI on a number of pharmaceutical grants that investigate aspects of atherosclerotic cardiovascular disease. When I asked him about his proudest scientific impact, he said it was through his mentorship of numerous physician scientists and PhD scientists and RD PhD scientists. In 2019, he was awarded the Outstanding Mentor Award for Colorado's NORC, their nutrition obesity research. Among his major scientific contributions is his being one of the first to show that the brains of individuals who are lean versus those with obesity reacted differently in fMRI when shown pictures of food. And this is the topic we'll hear about today. So please welcome.
All right. I think, do we have people online? Do we need to use the microphone for that?
Yes.
Okay. All right. Great. Well, thank you, Elizabeth. That was a very nice and warm introduction. Yeah, I get to follow Dr. Parks. You know, she's past president of the Obesity Society. I'm the incoming. So she knows. She feels bad for me. So what I thought I'd do, especially since I knew there would be a lot of students and such here, is kind of give a story about how my career kind of went through its kind of path. And so we're going to do a little bit of background, but then we'll get into some of the early work that then drove me to get to doing brain imaging work. That's kind of be the goal of this discussion. And again, if you have questions, whatever, I'm not a formal kind of guy. Just ask, just raise your hand or blurt it out or throw things at me, whatever you want. But let's go ahead and just jump into it. Let's see. Sometimes you have to click on. All right, I have some disclosures that are not relevant to this discussion. I'm a clinician, and as I told Elizabeth last night, one of my favorite things to do every week is to go in the clinic and see patients. You know, it's wonderful to have that. It's an honor, let me just say that, to be involved in somebody's health and their care. So I have to put this into context of some cases. And the first is a pretty typical one that we see in the weight management clinic. It's a 36-year-old woman. She wants help with weight loss. She reports slow, gradual weight gain over the past 10 years. This was especially a problem after her 2 pregnancies, when she gained a lot of weight, didn't lose it all back, gained again. Most of her family members are overweight. She can't understand why she's gaining weight. My metabolism must be low, she says, you know, and we all know that's probably not necessarily true. She's tried every diet out there. She really has tried hard. She doesn't feel like she eats anything. I don't eat at all, right? And you do a 24-hour diet recall, not a great tool, but not bad. And it suggests she doesn't eat very much, right? She says she doesn't eat out of hunger or out of emotion. She denies significant snacking or ingestion of liquid calories. She works out, you know, a couple, 2-3 times a week. What else can I do? She asks within frustration, and usually there's tears involved. And this is something we see all the time. So, a few questions come up when I see these patients. First is, she says she doesn't eat anything, right? And so, she has to be eating more than she reports. Otherwise, she wouldn't be gaining weight, right? She has to be in positive energy balance to be gaining weight. So there has to be more calories coming into the system than what she's describing. Is this her true perception? Does she not sense how much she eats on a day-to-day basis? Is this part of the disease? And I believe it is. She doesn't really know how much she's eating from day to day. She's not lying to you. She's not purposely saying, I'm not eating anything. She truly doesn't believe she's not eating anything. She doesn't eat out of hunger. And we hear this all the time. And so does she not feel hunger? Does she not get satiety, satiated? What's going on there? And this is a very subjective feeling. And so what does that mean? And again, why does she eat then? Or why does she stop eating if she doesn't feel hunger or satiety? And I would say, again, is this part of her disease? And then finally, why is she not adapting to the excess energy that she's in state? Why is she not down regulating? What's going on there? So are her homeostatic mechanisms, the central circuitry, not working properly? And I certainly think that is part of her disease. On the other hand, we were interested in thin people. I use that term instead of lean, because these were people that truly were thin and that were resistant to weight gain. And we did a study, and we had advertisements. Basically says, can you eat whatever you want and you don't gain any weight? And so this guy came in for responded to our ad. He's 29. He's always been thin and lean with a body weight that's never really fluctuated by more than 5 pounds. He enjoys eating good food as much as the next person. He eats pretty much whatever he wants. He doesn't really pay attention to it. He's never really dieted per se. He gets hungry and eats when he's hungry. He's also modestly physically active. His wife, on the other hand, struggles with weight keeping her weight under control. And yet they're in the same household. They eat very similarly. So he's interested in why he doesn't gain weight. And we had a lot of people calling us up for this study. It was actually pretty interesting. Even people from across the country, because it's made the national press. Is this genetic? What about his children? What's going on here? So again, some of the questions I ask myself when I see this guy is, why doesn't he gain weight? And that's, he's in the same obesogenic environment as our lady in the first case. Are his homeostatic mechanisms in terms of regulation of energy balance, are they just more sensitive to perturbations in the environment? Is he more able to adapt and compensate? Is it behavioral? Are his behaviors more conducive with dealing with the environment? So how, in fact, does he adapt? So we were interested in that. So we know that, again, I don't have to say this for everybody here, obesity is driven, rates have risen dramatically in a fairly short period of time. We're talking over decades, or maybe now 50 years. While that's, genetics are important, that's really a pretty short period of time for genetics to be the only cause. So we believe that it's genetics as a background, but really it's the environment that's the main culprit, with the abundance of cheap, good, high-fat food leading to increased food intake, new technologies. Our workforce has changed completely from more manual labor to sitting at a desk and doing service type, so less activity. And we certainly know that both of these things are going on. So if we look at our feedback mechanisms, they're primarily designed to protect us during states of undernutrition. And so the system isn't as well designed for dealing with excess calories and inactivity. So we asked the question, this was when I was still a fellow with my mentor, Dan Bestison. We said, well, why isn't everybody overweight? We're all subjected to this environment. Why aren't we all obese? So we were actually more interested in the thin people and how they kept their weight down. What was different about them? And certainly thought that was a good model. And he ended up studying this obesity-resistant rat strain. And I went in a slightly different direction, looking at it more on the human side. So here's the idea. This was proposed by Eric Ravison a long time ago. If we're all exposed to the same environment, why are we not all obese? Some individuals must adapt. And so, what we see is that you have individuals who are either resistant or prone to obesity. In the old restrictive environment, essentially nobody was obese. But then you get into an obesogenic environment like we live in. Those people who are prone now have increased BMI. That's kind of the idea, the interplay between the genetics and the environment. Now, if we look at rat models, we see that they're really pretty good at adapting to the environment, right? So you have a rat and you feed it and it keeps growing at this rate. That's the blue line here. If you overfeed a rat at this time point until here, they gain weight. If you now give them access to free and limited food intake, what do? They lose the weight and they're right back to where their natural trajectory would have been. Do the same experiment, but now you underfeed the rats. They lose weight. Now you give them free access. They regain the weight right exactly to where they would have been. So that really gave us the notion of a set point. The question, does that happen in humans, right? So do we adapt at all to the environment? And I think there's a few examples that suggest that we do. So if you do overfeeding chronically, we see that there is less weight gain than would be expected for the amount of calories ingested. There's significant inter-individual variation in weight gain. So if you give everybody 1,000 calories more, some people are going to gain more, some are going to gain less. So what's going on there? And the two, classic studies the first was the Bouchard twin study published, eons ago for the young people. I guess you got some of you weren't born yet, were you? And they gave 84,000 excess calories over 14 weeks. These people lived in a camp, basically. And the expected weight gain based on calculations that they should have gained about 11 kilos. The mean weight gain was only 8 kilos. Right? So there seems to be some adaptation. The range, though, was anywhere from 4 kilos only to 13 kilos. So more than the expected weight gain. So it's definitely something going on. And then there's the Levine study out of Mayo called the fidgeting study. which was, really a hot study in 1999, 56,000 excess calories over 8 weeks. I think it was 1,000 calories a day. And then the expected weight gain would have been over 7 kilos. The mean weight gain was only 4.7 kilos. And look at the range. There's one person that only gained 1.4 kilos, despite 1,000 more calories every day. And one person lost, and it ranged all the way to somebody gaining the exact expected amount of weight. And in this, basically, they said, oh, it's about non-activity thermogenesis, which is explaining these differences. So it seems to be something's happening in terms of adaptations. So I kind of thought about these studies and how people were dealing with the environment and how are we adapting? And certainly we can increase our energy expenditure. That's what the Levine study would suggest, that it's on the activity side that we, through non-exercise. But certainly some people might adapt by being more volitionally exercising, so true activity, right? We don't think thermic effect of food is having a big effect, and resting energy and expenditure really doesn't predict that either. Could be differences in substrate metabolism, so changes in fat oxidation, how we assimilate nutrients, the gut microbiome. There might be adaptations there that impact this. And then finally, it might be on the energy intake side. People might adapt by eating less and having changes in their appetite, which leads to changes in behaviors, et cetera. And this is what we wanted to study. So this is, we all know this. I will spend only 2 seconds. We know there's a complex interactions of different signals, long-term adiposity signals that either enhance food intake or suppress it. And we have short-term signals as well. We know that these long-term signals really are leptin and maybe insulin in the brain. But we know that the gut's involved here with ghrelin being a stimulating hunger, GLP-1, PYY inhibiting hunger and promoting satiety. So a complex system at the homeostatic level. And I don't want to spend any time on this more because you guys know all this. But if you think about it, if it's all driven by the physiology of this homeostatic regulation, you overeat, that puts you at positive energy balance, you gain weight, you gain fat mass, you make more leptin, you lean body mass gain. The feedback mechanism should go back and tell you to stop gaining weight and to return your weight back to baseline, just like our rat models with that set point. But there's something that's broken here, right? Something that's not quite working right in most individuals. So what could be some of the issues with our homeostatic or biologic signals for food intake? It could be that these signals are mainly designed to protect us during times of undernutrition, and so the system just can't handle overnutrition. Perhaps we develop resistance, so resistance to leptin, for example, or to other important signals, and that certainly may be part of the story. Or maybe it's just a heck of a lot more complicated than that. And that's when we start thinking about, well, what else is affecting our food intake? What are the non-homeostatic regulators? And I, borrowed this from a number of different people, but this has been my model over the years, is that one is there are internal brain inputs, reward mechanisms that are important, cravings relating to reward. Just the cognitive thinking about food is important. Restraint, so actually cognitively restraining yourself. Learned behaviors are important. Attention is very important. And then you have these external inputs. You have the environmental cues, the sight of food, the smell of food, the taste of food. These are all critical. Availability and portion sizes are important. We know that the bigger the portion, the more people eat. The more available food is, the more we eat. You would not have eaten any pie today, I bet you, if there wasn't pie sitting on the table there, right? It happened to be available. You saw it, you smelled it, then you tasted it, and it was good, right? Okay, so that's outside of our homeostatic mechanisms, right? I mean, some of you might have said, no, I'm not eating, my PYY levels are high, I'm not hungry, I'm not going to eat. That's certainly possible. Social context is important. Again, you were forced into eating that pie because it was socially acceptable to do it. But you might be in another circumstance where you don't eat because of social reasons. Time cues are critical, right? We know because of the workforce, we were forced into eating meals at specific times. And that all happened with the Industrial Revolution of you got to eat breakfast, most important meal of the day, according to Kellogg, right? And then you had to eat when they gave you a break midday. So that was lunch. And then you came home and you were hungry and you ate dinner. So time cues are very important. It's 12, I'm hungry, I'm going to eat, right? So all these things have to be assimilated by the brain. So again, my model is that you have the homeostatic mechanisms, which primarily are working through the hypothalamus and other central regions in the brain that lead to energy intake or reduced energy intake. But then you have all these non-homeostatic mechanisms, the internal, the external inputs that obviously are communicating. And these can directly tell you to eat or not eat, right? Just like the pie. Your homeostatic mechanism said, don't do it, but you did it anyways. Thanksgiving, same thing. There was pumpkin pie out there, right? It makes me think of Thanksgiving. You're full. You just ate a table full of food. And yet here comes the pumpkin pie and you still eat it. That is direct non-homeostatic. And then what we'll show is that there's interactions between these things, right? Interactions between the internal non-homeostatic and the homeostatic via higher brain regions, maybe even through central regions like the hypothalamus. And it's my belief that these mechanisms are overriding the physiologic signals in many of us. All right. So when I was interested in studying this, we said, well, how can we study this in humans? In rats, it's a lot easier. But in humans, what can we measure? We can measure eating-related behaviors, and those are important. Well-validated questionnaires. We can actually measure whether people eat. That's the ultimate behavior, right? So behaviors can be measured. We can ask about appetite, but that's really subjective. What hunger means to one of you might be different than to another. Satiety might be nausea to you, to something else to somebody else. But we can measure these things, and there are validated tools. We can measure energy intake, but how accurate is it? If it's in the lab, it might be accurate, but that's not real life. If it's free living, how do you know the person really ate what they said they were going to eat? We use pictures, we use way back measures, things like that. But we're always wondering, how accurate is this? We can measure hormones, metabolites. Oh, we can measure ghrelin, PYY. They're commercial assays. It's easy. But that's only telling you what's happening in the periphery, in the blood. It's not telling you what's happening in the brain. So again, how important is that? I think is a question. All right, so one of my first studies here's my mentor, Dan Bessison. Back when he was my mentor, initially he had black hair. He always kids that he's a gray-haired guy now. He's a past president of the Obesity Society a few years ago. So we decided to do a study where we were going to give people excess food. We were going to put them in that environment. Short term, 3 days. We felt like 3 days was enough to get a signal, yet not long enough to cause a significant change in their fat mass, which could have independent effects. So we said 3 days. And we're going to study this in people who are prone to weight gain and people who are resistant to weight gain. And we were going to measure all kinds of metabolic things. We did insulin clamp studies. We were looking at fuel metabolism. We were going to do all kinds of crazy things. But on the side, we said, let's measure hunger. And I talked to somebody about that today. When do you add things that you aren't really that interested in? As long as it doesn't add a lot of subject burden, easy to do. So we just did, these little visual analog scales and said, how hungry are you? And it was pretty fascinating. So we had a eucaloric period where people were given a diet that was supposed to keep them in energy balance. And then we had an overfeeding period for 3 days. And we'll see on the left of that dotted line is the hunger scores. We had the thin obesity resistant people, and then we had the reduced obesity individuals who are obesity prone model. And you see their hunger scores are essentially the same. If I did satiety, it'd be the same. Then we started overfeeding. And what you see is the black dots is within a couple of meals, their hunger ratings dropped dramatically. And if I showed you satiety, their satiety ratings went up dramatically within a meal or two. But look at the reduced obesity patients, individuals. The first 2 full days, there was absolutely no change in hunger. And again, with satiety, there was no change in satiety. It wasn't until the 3rd day we started to see a drop off. We were fascinated by this. What on earth? 50% more calories is what we gave them. And yet it was like they weren't feeling it at all. And we would interview them And the thin people were like, oh, the food's up to here. I feel terrible. This is awful. The people with, obesity and reduced obesity were like happy as clams. It's like we weren't even overfeeding them. So it was quite fascinating. And so that's what drove us to say, well, what's controlling that? And how can we study that? I'm going to skip that. We looked at some homeostatic kind of mechanisms. We said, okay, how is leptin playing a role in this? How is ghrelin, PY, those kind of things. Liz Thomas was a postdoc endocrine fellow that worked with me and now has her own R01. I'm so excited. And she analyzed some of these data. So she didn't do these studies, but she analyzed these data when she joined me in my research. And what we showed is that people who are obesity prone, they already had higher leptins, yet they weren't more full. So that's telling you the leptin's not doing its job, right? Ghrelin levels were higher in the obesity-resistant people. Those bars don't look that different, but it was statistically significant. And that's been shown over and over again, that lean people have more ghrelin. Ghrelin is supposed to be a hunger hormone. So if you have more hunger, maybe you should be bigger. You should have more, be eating more. But no, actually we see the opposite, suggesting maybe people with obesity proneness have a resistance to ghrelin potentially. And then we looked at hunger, and hunger was no difference between these groups. This is that kind of baseline, suggesting that there's something wrong with the signaling due to these hormones. So then we said, well, that's great and all, but what we want to know what's going on in the brain. We can't do brain biopsies. We can't, get samples. So yeah, we could try to get CSF. We talked about that, but I didn't want to have to do LPs and learn how to do that. I didn't become a neurologist for a good reason. So we said, can neuroimaging help us? And this was at a time where fMRI was just starting to be a tool that people started talking about. And at our institution, our Department of Psychiatry had gotten a new MRI scanner for research. Their interests were substance abuse, schizophrenia, and they sent an email blast saying, we have a new machine, come to our meetings, we want to collaborate with people. I'm like, okay, I'll go. I went and I talked to the head of the department and he said, oh, you talk to Jason over here. He'll help you design a study. You guys should do something. And that started a collaboration that we still today are working together. So Jason Trigalis, if you ever look at my publications, on every paper of mine, right? So he's the brains behind, no pun intended, behind the brain imaging work here. So what can you do with neuroimaging in research? We can look at anatomy with CT, MRI. We do look at structure of the brain. We can look at neuronal activity. That's more interesting. We can do that basally or stimulus related. And there's different methodologies, but functional MRI would be the most effective. You can also use receptor or metabolites and peptide activity, using tracers, using PET technology. That takes even more expensive, even more invasive, more radiation, et cetera. So we chose functional MRI. All right, with functional MRI, you need a stimulus to light up that brain, all right? And so what we chose is to show images of food, the pie, right? So when you walked in, you saw that pie, what happened to your brain, right? I want you to think about that. But we could have also said the smell, but creating a machine that pumps out smells is very difficult. There are people who've done that. We could do taste, but taste eating in the scanner is a big problem because that you chew, that causes motion and that causes a lot of issues. We did some taste studies with liquid, with sugar, but that's certainly another option. We can have people think about food, and there's been some studies doing about that. We can do other tasks that relate to eating behaviors, things that relate to attention, impulsivity, et cetera, and we've done those kind of tests as well. We chose to use visual cues. To know a food is available, you have to see it first. To know it's not toxic, to know it's palatable. So we feel like the visual is really the number one cue that tells you to get ready to eat. You usually see it before you smell it or taste it. We're constantly bombarded by visual cues. That's why the food industry advertises so much, right? It works. Ultimately, it's easy to use in a scanner. And that's the reality, right? And it's very well validated now, especially after a number of years. These findings are very reproducible within our own hands, within the across different research groups, and the findings are quite robust. This was a meta-analysis, and then some of our data were included in this that was published, oh God, it's a while ago now, 2011. And they found that within all these studies, there was this network of brain regions that activated when you were looking at images of food, specifically highly palatable, what we called hedonic foods. And that included, orbital frontal cortex, which is more prefrontal, telling you, thinking about it. Posterior cortex, more about attention, which makes some sense. And an insula. Insula is going to pop up over and over again. And insula has a lot of potential effects. Anterior insula is more about taste receptor, but the posterior more about other things like interoception, trying to understand your body's signals, memory of food and taste, and motivation to eat is a big one as well. And you'll see this come up over and over again. So that's what the grouping with the kind of usual network. So here's Jason. Now, he didn't have a beard when I first worked with him, but now he's got a beard. And so we did our first study, and we started by looking at what's the normal response. So we took thin people, obesity resistant. We thought that would be the most normal response potentially. And we had people look at images of food that were either highly palatable or not. And then we compared that to objects that were not food related. That's similar brightness and colors and that kind of thing to take out that kind of noise. And so you're always looking at what part of the brain is more active when you see a hedonic food versus a chair. Okay, so that's how we think about it. So you have this contrast. And what we found is this network of brain regions, again, similar to that meta-analysis, but ours being just our study, we see a few more things pop up. So that insula is there. Let's see if I can use my arrow here. We have sensory cortex. Interesting. Ventral striatum. Well, we got into reward networks, which makes a lot of sense. Hippocampus, memory regions. Parietal, we talked about attention. Inferior visual, that one always baffled me. That's your visual cortex. If I'm looking at that chair, my visual cortex is activated. But when I look at the chocolate cake, it's more activated. So again, maybe attention related to the images, telling you that it's highly palatable. We've done these studies over and over again, and we see pretty much the same thing. So again, these are brain regions that are important: attention, reward, motivation, memory. And this is what's happening when your brain is hungry and you're looking at all these yummy foods. Now, what happens if I overfeed these people? Remember, these people are lean, normal weight individuals. And I give them, in this study, we gave them, I believe, 30% more calories. We reduced the overfeeding a little bit. This is the image. I mean, all that went totally away, completely gone. This is fasting. They haven't eaten breakfast yet. There's absolutely no activation. So it was pretty crazy. And in fact, there was reverse activation. There was inhibition of hypothalamic activity, which I don't know what that means at the level of this brain type imaging. I can't tell you which neurons are being activated or inhibited. So pretty interesting. And that was kind of our aha. The brain is very sensitive to these signals. Then we acutely fed these people. And what happened? You turned off. This is inactivity of these key regions like the inferior prefrontal cortex and the insula. So acutely eating turns these things off, but even 3 days of overfeeding turns these off. All right. So then we want to know, is it different in the people who are prone to weight gain? And this is like the ha-ha moment. This is it. If you want to leave after this, you can go. All right. This is my one big slide. So we looked at people with reduced obesity, because that was our model, because it's hard to predict people if they're going to gain weight or not. And we compared them to people who had obesity resistance. This was defined through family history, no history of weight gain and dieting, et cetera. Reduced obesity individuals lost, I think, about 7% of their body weight in a 3-month period of time. And then again, we're comparing hedonic foods to basic objects, I should say objects. Well, I want to make sure you have the contrast right. It's a double contrast. It's two different phenotypes, two different kinds of images. All right. And so this is what's happened. So here we have the pretty pictures, but we have this quantified. And so what you see, so we have the thin people in eucaloric state, their insula is turned on when they're hungry, and then we overfeed them and it turns off. In the reduced obese individuals, their insula actually is less activated. And what happens when we overfeed them? If anything, it went up, but there was absolutely no change. We looked in the hypothalamus and visual cortex, and we see the same effect. And so it was basically a complete opposite effect. And this is with 3 days of overfeeding. So pretty dramatic, and this correlated to those effects that we saw in the hunger scores and the satiety scores, where there was very little effect after a few days. So that told us that really that the neurocircuitry in these individuals is very different. And are they born that way? Is it learned? Is it through like gliosis in the hypothalamus? I don't know, but clearly it's altered circuitry, which again I think is more evidence that this tells you that it's a disease. Yeah. They were age matched. They were all fairly young people. So then we were interested there's a new area of fMRI, which actually took away the stimulus. It's called the brain default network, or others call it the resting state brain. So when you're sitting there doing nothing, your brain is activated. And it's these three main brain regions: the medial prefrontal, the posterior cingulate, and the lateral parietal. Why those regions? We don't know. But we know that this is across many studies, many people. And so we decided to look at this. This was a kind of a new finding in the literature. And lo and behold, well, here, this is the thin individuals, the obesity prone, and then you had the reduced obesity individuals below. So they both have this area turned on when they're not looking at pictures of food. But when we actually quantify it, we see that the people with obesity proneness had more activation in these brain regions. So there was more of this activity. And then when we overfed them, did I show this picture? Oh, I didn't show it, sorry. They weren't able to turn off this resting state to the same degree as the thin individuals. So it made us think, are these patients more engaged in or greater attention to their internal state? They just can't shut it down. Are they less able to focus on others? Because if you can't turn off your resting state, you can't focus on the other stimuli. And so this could be part of the problem, part of the disease. All right, so that was great. So this is all very descriptive physiologic stuff, but I think interesting, and we were excited about it. We said, can we do anything about this? When I went to medical school, I was told the brain was hardwired and was not plastic and could not change. And we've known now that that's not true. The brain is plastic. And so could we do anything about this? So this study, I don't remember where these folks are. Deckersbach must be, it's not who knows where in Germany. They did a behavioral intervention to see if that altered the food cue responsivity, and they did show. It's not robust, but they did show a change, suggesting even with a behavioral intervention, we might be able to alter the way you're looking at these food cues. So a first step. What about appetite-related peptides? And there's been a number of studies looking at that. I'm just going to summarize them. Leptin. So if you look at people with leptin deficiency and you give them leptin, you completely restore the brain activity in the brain. This includes the stuff that we talk about, the insula and the prefrontal cortex, et cetera. So these patients, I mean, these small numbers, we're talking 3, 4, 5, 6 patients that they've done these studies in, but their brains just light up like crazy, and then you start them on leptin and it all goes away. Group out of Columbia did a study and looking at giving leptin back to people who had lost weight. So their leptin levels went down. So now they give them leptin to see what happened. And again, giving them more leptin. Now that they're reduced obese, maybe they're more sensitive to leptin, and they were able to alter the signal to visual food cues. PYY, GLP-1 experiments have been done acutely, and again, have shown to impact brain regions such as the insula and in reward processing. We've seen similar studies done with ghrelin. Again, these are all acute studies suggesting that these peripheral hormones that we all think work in the hypothalamus and the brainstem, maybe in reward center, they're impacting brain regions that are higher brain, prefrontal cortex, insula, parietal. And now with the GLP-1s, we know that there are receptors for GLP-1 in all these brain regions. So that was quite interesting when we saw this. It wasn't just about the hypothalamus. Then we started thinking about exercise. We said, well, has anybody looked at exercise? And there was a couple of studies done, 2012, 2014, that did an acute bout of high intensity exercise to see what did that do to the brain response to food cues. We know an acute bout of high intensity exercise tends to reduce hunger, make people not feel as good as it relates to eating. And sure enough, these acute responses did alter the brain response to food signals. But that was just a single high intensity exercise bout. So we decided to do a study. So again, with Jason on the left, and now collaborating with Ed Melanson on the top right, who's an exercise guy. He was doing a study looking at a 6-month aerobic exercise intervention. He was interested in exercise adaptation, to activity adaptation, et cetera. And I said, Ed, can we do some brain imaging studies in your subjects? He's like, I have 12 more subjects to do. Absolutely. So we did some pre and post. No control group. It was just pre-exercise. 6 months later, we repeated the test. And what we know, the exercise, was associated with a variation in weight loss. Some people lost a bunch of weight, some gained, some didn't lose any. The average weight loss was about 2.5% in that cohort. And what we found is that not in the setting of acute exercise. These brain imaging studies were done at baseline and then after 6 months of exercise, but not on an exercise day. So this represents more of the chronic effect. They might have exercised I think we gave 2 days. They couldn't have exercised for 2 days before these scans. And we found a significant reduction in the activity. And again, these similar brain regions, the insula, the parietal, the visual cortex, suggesting that the exercise was having a significant effect on how people were viewing the food, at least. And that was associated with the weight change. So the more there was a reduction in insular activity, the greater the weight loss. Now you could reverse that. Say the greater the weight loss, the more reduction in insular activity. Well, chicken or the egg. These are associations. It's not. I can't tell you that it's causative, but certainly intriguing. Based on these preliminary data, which again was only in 12 individuals, uncontrolled, we still were able to get it published. So just keep at it. When you're having trouble publishing papers. We were able to get a couple of grants funded to do this work. We've now done several exercise studies, and we have not yet published the imaging data. So I don't have anything to share with you today. The work is still being analyzed. We just finished these data, collecting these data right when I was leaving Colorado. 2 years later, we should be done, but they're slow at getting. It's a slow process. So we have a couple of papers now. And so we had an aerobic exercise group, a diet group, we had a resistance exercise group. And so we're very excited about this, but I don't have anything to share with you yet today. Stay tuned, I'll come back. One thing we were interested in is if you got a good response to exercise, you lost weight, were you more likely to have had a benefit, in terms of reducing the insular activity? And their answer was yes. Again, we were never able to publish this. We were able to use this for grant purposes. There was just too few people in this responder versus non-responder subgroups. But everything went in the direction that we would have expected. Then, we showed you the default network, the resting state. So this is Christina. At the time, she was McFadden. Now she's Leggett. And she came into the lab as a postdoc and then got a K award. And she looked at our exercise data and she found again that the exercise, quote, improved. It reduced that resting mode network or that resting state activity, which we think would be a good thing for these individuals. And that was a pretty robust change in those activities. We'll go through that. What about medications? Now, we haven't done this work, but this is published from some other groups. This is with liraglutide, which is a short-term daily injectable GLP-1 receptor analog. And these images are small, but the idea is that, yes, these brain regions in higher brain regions are being affected by this GLP-1 receptor analog. Not a lot more data. And we know in animal models that there are GLP-1 receptors all through the brain, and these are impacted and activated by GLP-1 given peripherally. Naltrexone bupropion is sold in the United States as a drug called Contrave for weight loss. And again, was studied and showed impact on the brain response to food signals, all going in directions that would make sense compared to what we described. Bariatric surgery. There's been a number of small studies, and I just kind of throw these up there just to say, again, consistent effects. We have some unpublished data. We just never were able to get enough patients who completed surgery to do our study, but we certainly saw this effect of the surgery in the short term causing these pretty robust changes in the brain. And then we have this interesting study that Christina put together. It's our implicit priming study. Again, can we alter the brain with different interventions? And so what is implicit priming is we're pairing a disgusting image with a highly palatable, high caloric food image. And in the first study we did, we showed that we could reduce the liking for the food just on a questionnaire. How much do you like this food? How much do you want to eat it? Those values dropped pretty significantly when you paired that food with a disgusting image. Now, disgusting image is not seen. It's like subliminal advertising that became illegal, right? It's 0.2 milliseconds that it's shown for. They don't get. We asked. Did you see this? And they don't ever see it. And these disgusting images can be images of horrible, traumatic car accidents or gunshots and or nasty bathroom stuff, really stuff that it's boom, you don't see it, but it's paired with that image, yes. And so then we repeated it and did it neuroimaging, and here is an example. So you have kind of the neutral, you've got the disgusting image, and then you got the pretty food. And what we saw is that led to a reduction. This was acutely done, a reduction in image, that was the ratings. And then we saw pretty robust changes in the brain based on that. So now we have, Christina was able to get an R01 that I'm a co-investigator on, where we're doing this as a chronic intervention to see if we can make this effect last. Now, we tried to do the opposite. We tried to pair kind of healthy food, like vegetables and stuff like that were kind of rated neutrally with like something really a good thing, right? And this actually made some New York Times or something article. They called it the kitten study or something. Cute pictures of kittens and bunnies and things that make you laugh and smile. It didn't work at all. Now we piloted, and we've never done the true experiment of doing sexual pictures. That worked.
To reduce or
to improve the liking of the healthy foods. We just piloted it in a few people, but we never actually did that study. So pretty interesting. So this, again, I don't think we're going to be doing this as necessarily as an intervention to treat obesity, but it tells you that we can alter the brain through other ways. Yes.
With altering mechanisms, did you measure actual? Have you tried to measure
that's what we're doing now with the chronic. Yeah. So we don't know yet.
Isn't there something that if you put a green light on food that makes food look moldy, people won't eat it? If you
similar,
so
right?
Maybe a disgusting picture is somehow heightening aversion or some weird thing.
Yeah, absolutely, absolutely. So I want to have time for questions. So I'm going to summarize some of the things. I'm going to actually, I'll probably just read this to you. So clear differences in the neural response and sensitivity to food cues and changes in energy balance in individuals prone to weight gain compared to those not. I think that's pretty clear. these alterations in the neural response are associated with structural. I didn't show you those data, but we've shown structural changes in the brain and that resting state that we discussed. So I think that's the key. We think that this altered neurocircuitry in patients is likely an important underlying cause of their overweight and obesity. You could say it's potentially just associated, but certainly the fact that we can reverse it with different interventions suggests that it might be causative. So I think we say good news that the brain is plastic. There is plasticity to these responses. And then these to interventions like exercise, the pharmacotherapy, I think, is a big one, and surgical interventions as well, and maybe others. Elizabeth and I were talking about this last night. Is this a normalization? And I don't know what normal is, right? It's hard to define that, but certainly the responses are now more like someone who doesn't have obesity. So I would say it suggests normalization. Maybe that's how some of these drugs are working. And maybe, the question is, how long do these effects last? What we've not done is repeated images 3 months after the exercise intervention is done to see if we have continued effect. My guess is no, but again, we don't know the answer to that. I didn't discuss it because we didn't have time, is that there are sex-based differences. And I've had a lot of conversations today about different, a number of you looking at sex-based differences. There's clear differences in the response to eating behaviors, but also we have shown in some secondary analyses of our studies that there are sex differences in the response to food cues. And what we're doing now is, we have an R01 to look at this in a systematic way. That study is still ongoing. We know all the challenges of doing these kind of studies, having to set up these meal studies and imaging days in the follicular phase. Damn, that's hard. Those women don't cooperate. And then we do it again in the luteal phase. That one's a little easier because we can time it from their last period, at least get to that right area fairly, a little more accurately. And doing it in women who are not on birth control pills, who are not on hormone releasing IUDs, they're not, it's not an easy study to recruit for. So it's taken a little longer than we expected. And we're comparing that to men. So age matched, BMI matched men. And these are overweight individuals with mild obesity. So I think that's really important. We're measuring sex steroids and such to look at potential mechanisms, that kind of thing. So that's a work in progress. All right, and I'm going to, in the last minute before we get to questions, I'm going to come back to my 2 cases. Does this help me in my clinics at all? And, if we come back to our lady who doesn't feel like she's eating anything and struggling, a lot of this tells me that, her genetics, all the family's overweight, in her biology, maybe there's fetal programming involved in this because mom was overweight during her pregnancy. It favors nutrient storage and excess calorie consumption. So how do we alter her biologic signals? I don't know that we can't change her genetics. We can't change the fetal programming. We can do it for the next generation, maybe. And so this, tells me that I need to treat this disease and potentially alter the neurocircuitry with pharmacotherapy. And I think she meets the criteria for it. And certainly that's one of the reasons to consider medical therapy for management of overweight and obesity. So I think that's our best way of handling this part of the disease. Now, some of her disease may be, and again, this might all relate to her genetics and biology, is that she's less sensitive to environmental cues, and that's, I think pretty clear. So how do we alter the environment? That's hard to do. Do we look for strategies, so more behavioral strategies to help her interpret her external signals? Does mindfulness, and we've done there's some studies being done with mindfulness and that kind of thing, where maybe she can pack that resting state to a point where she can turn it off and then be more sensitive to the environmental cues around her. So that's, I think, a direction for behavioral therapy. Look at techniques to help figure this out. Now, I would argue that medical therapy might help her with this as well. We've talked about the noise, food noise, right? And that maybe these medicines help reduce that and help her be more able to adapt to the environment around her. Now, one could argue that it's just behavioral. I don't know what that means, that her behaviors are at fault. So if that's the case, again, maybe drugs might help. Look for better strategies to facilitate behavior change. And then obviously we want to know underlying, is there mental health issues that we need to treat? Are there emotionally related behaviors that are impacting her food intake? Shift eating disorder that's impacting. So these are all important things to focus on. And then for a guy, I'll come back to you in a minute. So he can eat whatever he wants. He's thin. My wife yells at me for being like that. Although this year, Charleston, had good food there. His biology may be more sensitive, it allows him to be more sensitive to perturbations in the environment. He's more like the rats, he overeats for a few days, but then he goes back to his usual diet or usual activities and the weight comes right down, So he's able to do that. Maybe he has that set point. He might be more sensitive to the cues around him, or maybe his learned behaviors are different if there's a family component there. So I think the most important thing with him is say, good for you, way to go, keep it up. But what can we learn from him? And that's what we were trying to do with our study. So acknowledgements, most of this is all done in Colorado and still ongoing in Colorado. I'm not doing any of this work at MUSC just yet. We'll highlight Jason Tregellis and Christina Leggett for my primary brain imaging collaborators, and Dan Besson, who is my longtime mentor. And then there's lots of other people, and then the fellows and students who did a lot, and all the other and the funding. So with that, I will end, and we have some time for some questions and discussion. Jill, you had.
You were showing case one at the end. So, okay, let's say you put him on a medication. Is this going to possibly change her perception of what she's eating? Like, she's telling you she wasn't eating much. One of the questions you asked her is, she did not realize.
Right. Well, that's a good question. And the answer is I would say probably yes, that now she's more, because we haven't measured that specifically, but our experience in the clinic with patients is they are very proactive now about, I know I can't eat that, so I'm gonna cut a little piece and eat that. So they're very conscious about what goes in their mouth, and they know, because if they overeat, they're gonna feel sick, right? So that certainly is, and I think some of the restrictive components of bariatric surgery kind of do a similar thing. So I think that the answer would be yes if we looked at that in a more systematic way.
I want to add, so I've gotten in these discussions about what nausea is, and the psychologists I've talked to, that if you start Thanksgiving dinner, you feel pretty good, you eat, you overeat, you will eventually feel nausea. You overeat, weight, and they say that's satiety. And I think of satiety as feeling warm and cozy because
Right.
I just had a little hot. But they say it's all one continuum, and so they don't overeat because they feel uncomfortable, but that is considered extreme satiety.
Right, and
Weird.
And that's a great point about satiety versus nausea. I think, I'm not a
Yes.
animal scientist, but certainly in the animal world, they call it aversion, right? And that is separate than satiety or satiation when you measure it in preclinical models. And the thought is that they are actually two different constructs, but I think it's hard to separate it out. And certainly, the most common side effects of GLP-1 receptor agonists is nausea. Yet most people stay on the drug, right? Because again, they learn what makes them not feel good. But I think nausea and satiety are intertwined. There are people who try to argue that they're very different things, and that some things will give you pure nausea, and that others, it's a society. I think it's so subjective, and that's part of the problem with doing the visual analog scales. I've had some people who feel like hunger is nausea. Like when they're so hungry, I feel nauseated, right? I'm like, okay, that's a very different feeling than what I feel. I feel like a hole in my stomach. I don't know. Everyone describes it differently. And that's what makes it challenging. And I skipped through a slide for time's sake. We looked at, and other people have looked at this. If you measure hunger in people, then you measure food intake, they don't correlate at all. Yeah, they don't. So hunger doesn't drive us, and people call ghrelin the hunger hormone, but I don't know if that's really why people eat if they're given, or animals eat when they're given hunger. Given ghrelin, or inhibited when they block ghrelin. So, yeah, so that's an interesting one.
I heard a patient say that they didn't go have ice cream with their friends because they knew going in they wouldn't enjoy it as much. Now, that's not about starting to eat and feeling full right away. That is blocking the anticipation of joy.
Correct.
They even know ahead of time, which is very
Well,
strange.
what also makes me wonder too is this associating nausea with being full. Is this something that they learned inappropriate when they're young, or was this something that's just?
Right. Good question.
Yeah.
So I'm interested in the applicability of the implicit priming. I can see how you can improve the ability of foods you are trying to promote, like fruits and vegetables. But I'm thinking about the opposite direction where you are trying to make the highly palatable food less appealing,
Right.
or less likable.
Right.
How does that translate in because as a mom, for example, if I had ice cream, I wouldn't want to pair it with something else.
Well, the question is,
Wouldn't want to.
could we do an intervention where you look on your iPad, once a week, and you're looking at all these images and you flash the nasty stuff, and once a week is all you need to train your brain? It's possible. Yeah.
I have a question about the asymmetries in the image from the functional device. Are those artifacts, or are those any meaning or differences between obese and non-obese? So some images clearly they have just a left light on the right side with.
Right. So asymmetry from the different sides of the brain. Yeah, that's a excellent question. And some of it is, I show a picture because it's pretty, but where we do the slice, I could have put that slice, a millimeter back, and now you would have seen more of the other side, maybe. So when we actually quantify it, there's less laterality than that, but there is some. That's a great observation. And what does that mean? I don't know. I don't know what that means. It's an interesting point. But most of the time we see the effects bilaterally. It's just the images are not, we try to just pick the prettiest pictures to show for our papers, and that it doesn't tell the whole story. That the more important from our studies are the tables that have all the coordinates, and when you see that, look at that, you'll see that most of it is bilateral. Couldn't hear you, sorry.
Insulin, differences in insulin response.
I have not looked at that. There's people who certainly are doing that with intranasal insulin, and certainly that's a hot area potentially. Can we block the fact? I think there might be one imaging study out there at least that's an acute effect of insulin in the brain, and I would imagine that we know insulin is an anorectic hormone. We think of it more of as a chronic adiposity signal than an acute, but certainly could be. But we haven't looked at that ourselves.
So you wonder if this starts early in life, and
Yes.
seem relatively non-invasive, so are these studies done?
There are some studies done in kid, and you see it early on. So, yes, and so, I think one of the studies was actually in kids who weren't overweight or obese yet, but their parents were. And so are they at risk? And there seem to be effects already at that point. Yeah,
Thank you very much.
Sure
And
thing.
everyone